Hello, everyone. Thank you for joining us and taking the time to learn about some cutting-edge work being done in Huntington's disease research. I'm Dr. Sarah Hernandez, and I'm the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsors for this webinar, Neurocrim Biosciences and Unicure, as well as everyone who has donated to the HDF to support the research that we fund, as well as all of you. Thank you for tuning in today. We have a really interesting webinar today that talks about using a new type of technology to study exactly where changes in the brain are happening in Huntington's disease. And it's easy to think about things in black and white or on and off, but everything exists on a spectrum. And the same is true for the brain and Huntington's disease. The brain has different locations within it that experience different changes because of HD. And there are also changes that happen in HD over time as the disease progresses. So maybe the level of one gene is high early on in a specific part of the brain, and then it is lower later on in the disease in the exact same part of the brain. And so if we can understand exactly what these changes are and where they're happening, it can really help researchers develop better technology, better therapeutics. And so this is a really exciting area of research because it's using new technology. So applying these cutting edge tools to HD research will hopefully get us to a therapeutic and treatment for HD faster. I am incredibly excited to introduce our speakers today because they have really been at the forefront of using these technologies for HD research. Our first speaker is Dr. Osama Al-Dalahmah, who is a neuropathologist at Columbia University Medical Center, York-Presbyterian Hospital. His research has focused on understanding how a specific type of cell in the brain called a glia is becoming abnormal in Huntington's disease. And so these glia are support cells that maintain the function of other brain cells. And ultimately, the goal of his research is to find ways to improve the function of glial cells to protect the brain from damage, to hopefully slow the neurodegenerative process. Our second speaker is Dr. Shawn Davidson, who is a Lewis Siegler Institute Fellow at Princeton University. Dr. Davidson research is at the interface of biotechnology and genetics, and his lab studies various diseases using something called imaging mass spectrometry. And this is a technique that allows him to pinpoint exactly where within the brain certain proteins are expressed. He combines understanding these changes in protein expression with disease-related pathology and mathematical modeling for the development of therapeutic strategies. Thank you both so much for joining us today and sharing your work. Before we start, I want to remind our audience that they can ask questions in the Q&A box throughout the talk. We have both HD families as well as HD researchers on the call, so your questions can be both technical or non-technical, just any question you might have. Send them in as the talk is going on so that you don't forget them. We're going to have about 20 minutes after our speakers for a Q&A session at the end. And thanks again for everyone for being here, and we'll get started by turning it over to Osama.
Thank you very much, Sarah. I'm going to share my screen now. All right, I'd like to first thank the Hereditary Disease Foundation for giving me this opportunity to share my work with you and communicate with you all and tell you about how mapping the brain is advancing Huntington disease research. All right, so a brief introduction. Huntington disease is a debilitating illness caused by a mutation in the Huntington gene. And a gene is essentially a stretch of genetic material that gives a cell the ability to produce proteins. Almost all cells have genes, and in Huntington disease, the Huntington gene has an error or a mutation. And that mutation causes the cell to make an abnormal protein, a mutant protein. And that mutant protein is sticky and it can cause aggregates. It can aggregate in different parts of the body and the brain, and particularly in a part we call the basal ganglia, which is involved in controlling the movement. But despite the presence of the mutation in all brain cells, the disease severity is not the same in all brain regions. We can see that using techniques like imaging techniques, which include CT scans and MRIs. Oops, sorry, I need to move this out of the way. I'm afraid I can't get rid of this annoying bar on the top, so if you excuse that. So when we look at these imaging modalities, an MRI of the brain, for example, here of a Huntington disease patient, we can see how the disease affects the structure of the brain. And from looking at these images, we can appreciate that the disease affects different parts of the brain differently. And in particular, this area outlined in the white box is particularly severely affected. This is a part of the brain we call the basal ganglia, and it's involved in controlling movement. We can appreciate the differences in how Huntington disease affects different brain regions when we do brain autopsy. And I'll walk you through this. So when a patient succumbs to the disease and donates their brain to research, we can do a brain autopsy where we look closely at the brain, both at the macroscopic level and the microscopic level. So what I'm showing you here on the right are slices from the brain that are divided into hemisections. So half of the brain is sliced in this way. And I'm showing you on the right side slices of the brain that were taken from an individual who died of causes that are other than neurodegenerative diseases. So we can consider this individual an otherwise controlled, healthy individual. And on the left side, we have slices from a patient who died of Huntington disease. And by comparing the two sides, we can appreciate the differences in how the disease affects different parts of the brain. Oops. So we can see that the disease affects different parts of the brain to varying degrees, and some parts are more severely affected than others. And again, we see that the basal ganglia region is severely affected. So in my line of work, we use traditional histologic techniques to visualize the brain under the microscope. We can take that part of the brain, the basal ganglia, and make preparations that allow us to image the brain under the microscope. And I'm showing you here a series of microscopic images that I stitched together to create this collage of images that includes the basal ganglia region. And in this region, essentially, we have pink parts and blue parts. The pink part has the neurons and other cells of the brain, and the blue part houses the connections between the neurons in this part of the brain and other parts of the brain. And we find variability in the severity of the disease even within the same brain region. So when we look at this part of the basal ganglia, we call the caudate nucleus, and look at the lower part of the caudate nucleus and magnify it, we can see that there are many neurons that are still there, and many of their connections that are shown in blue are still preserved. But if we look at the upper part of the caudate nucleus and magnify that part, we see that many of the neurons are actually lost, and their connections are likewise lost. So this tells us that the disease can affect the same brain region to different degrees. We can use traditional techniques to examine where the mutant protein accumulates. This is a microscopic image where we are trying to label the aggregated proteins. And we can see that the protein aggregates in different cells, and the aggregated proteins are shown in brown. So that little brown dot essentially has aggregated sticky protein. We can advance this further and look at the presence of multiple proteins at the same time. We can use one way to label one protein that is found in a particular cell type in green, and another protein that we want to measure in another color, and I'm showing you that here in red. So we can measure the presence or absence of specific proteins in specific cells. We can take that a little bit further and measure the presence of multiple genes at the same time. Again, using traditional techniques that have been around for many years. And here we are measuring the presence of 3 genes at the same time. Each dot represents one gene, and the genes are encoded by color. So the red dots encode one gene, the green dots that are shown here encode another gene, and the purple or the pink dots encode another gene. However, we have more than 20,000 genes in our DNA. To gain more insight into the disease pathology, we need new techniques to map the presence of all the genes at the same time. And one such technique that has become available in recent years is called spatial transcriptomics. And I'm going to introduce this new technique to you now. In this technique, we use a special slide that has these specially designed squares that we call capture areas. And each square measures 6.5 by 6.5 millimeter. When we look closely at these squares, we find thousands and thousands of regions that are covered by DNA molecules. And these DNA molecules serve as zip codes or area codes. So every part of this capture area has a specific area code. And these DNA molecules allow us to capture genes and assign them an area code so that we can map exactly where each gene comes from. So we can take a piece of brain and apply it to this capture area. And then each part of that brain gets an area code that allows us to measure the presence of all the genes in that part of the brain and measure it in a grid-like fashion. So every part of that grid will allow us to extract all the genes in it and trace it back to the exact area where those genes came from. The data looks complicated, but it can teach us about how different parts of the same brain region are affected by the disease. So in this example here, I took one part of the brain and analyzed it using spatial transcriptomics. And I can look at the presence of particular genes. And in this case, this gene, SYT1, is present in neurons. So I can measure the presence of that gene in different parts of that brain tissue. And I can do that for other genes. This other gene, CD2, is not expressed in neurons. And I could do that for all 20,000 genes. And by putting all that together, we can understand how the disease affects the presence of genes in different parts of the brain and different parts of the same brain region. And that allows us to make new discoveries in Huntington disease. The message I want to stress is a message of hope. There is hope with new technologies. The new research tools and technologies will allow us to make new discoveries that were not possible in the past. we're now applying these cutting edge technologies to human brain samples from people who donated their brains to research. This is very powerful because the discoveries we make from human brain samples are directly relevant to human disease. And I also want to stress that brain donation is important because it is the basis of the work that I do to learn more about how Huntington disease progresses and how to slow it down. These new tools hold promise for new therapies against Huntington disease. The new technologies and the new tools we are now using allow us to test new drugs against Huntington disease. But to test new drugs, we first need to apply them to Huntington disease models, which include animal models like Huntington disease mice. And we use those because they help us determine which drugs have the best potential to benefit Huntington disease patients. And one exciting area that we're beginning to research is metabolism in Huntington disease. And metabolism is essentially the economy of energy and nutrients. We want to ask questions about how Huntington disease affects metabolism in different parts of the body and even in different parts of the brain. And that is what my colleague, Dr. Davidson, is going to tell you about in his talk. Thank you very much.
During our live webinar, Dr. Davidson shared exciting new data that we've had to remove for confidentiality purposes. We are eager to share that data once it's published. In the meantime, you can learn more about Dr. Davidson's work by following the link in the description. Thank you to everyone in the audience who's been submitting your questions throughout the talk. Feel free to keep submitting those in the Q&A box. We'll get to as many of those as we can. Again, they can be both technical, non-technical. I know we have a mixed audience, and we'll try to get to as many as we can. So our first one is for Osama. For the postmortem brain slices that you showed, this is from Tacey Fox. Thank you, Tacey. The postmortem brain slices that you showed, did the individuals have a similar repeat number, a CG repeat number, such that you would have expected the 34-year-old individual's brain to look like the 48-year-old's when they're about the same age?
Well, this is a difficult question. So the slice of the control brain that I showed on the right was from someone who did not have Huntington disease, and the slice that I showed on the left is from someone who had Huntington disease. And as we know, the disease progresses at different rates in different patients, and that depends on the genetic background. This is exciting work being done on the Huntington disease modifiers, genes that are involved in modifying the age of onset or the severity of the onset. So the short answer is that I cannot predict.
Thanks, Osama. So this one is for you, Shawn, from Ron Barra. What is the significance of metabolism in HD?
It's a complicated question. I think that's what we're trying to really get at. So there's a lot of, there's clearly a lot of metabolic changes that occur. And I think what my work is trying to do is really identify which one of those is actually playing a causal role. And so I think there's interesting data on, just to pick one, ketogenic diet, certainly in different settings has, shown some modest efficacy. And so, yeah, I think that is the question. What is really causal? So the cure for Huntington's disease is, getting rid of mutant Huntington, but what can we do along the way to, significantly change the course of disease progression until we can get rid of mutant Huntington? And I think metabolism might play a role.
Thanks, Shawn. So this could probably be addressed by both of you. It's from Herwood Lang. And Shawn, you touched on this a little bit, but he asks, can the technique be used on brain bank matter, frozen embedded? What do you think, Osama? We will start with you.
Both. So we tried it with the frozen tissue. We have good success applying spatial transcriptomics to frozen tissue. And we've been applying it to fixed tissue. And for some cases that works better. So we use both.
Yeah, so we're the opposite. So the fixation and especially paraffin embedding makes things a bit more challenging for us. So we prefer frozen sections. We've actually started to look at what information we can get out of just formalin fix before you paraffin embed, but removing the paraffin is not a good process for us. So we prefer basically not to have paraffin embedded.
And this is another question that could probably be to both of you from Joshua Myers. Could you provide an example of a therapeutic that would benefit from this imaging, or rather how a therapeutic would benefit? And I think this is really important.
Yeah, well, we're both on a paper, so I'll start and then Osama. But one paper that we were just on looked at thiamine and biotin, and so we're actually looking at those two small molecules to see the localization of those in the brain. And we have a number of small. My technology will most benefit from looking at small molecule therapeutics. So anything that's a traditional drug, or a nutrient, my technology is best suited for. Once you get past, once you get at the peptide or protein level, there are better technologies suited to that. And we do a little bit of it, but that's not our focus.
Yeah, just to echo what Shawn has mentioned, these techniques are almost not only diagnostic, but they're so diagnostic of a drug effect. So if the drug can influences a particular cell population or particular brain region that we think is more affected than another brain region, then we can detect that with spatial techniques. But also allows us to nominate new candidates by comparing brain regions that are severely affected to ones that are less severely affected. And the work that Shawn is doing can be complemented with insight from gene expression. So we understand how metabolism is altered in different brain regions and how gene expression is altered in brain regions. And that together, that can give us insight about how we can leverage this knowledge to test new candidates and use the same technologies to test the effect of the drugs on gene expression and on metabolism.
Thanks, Osama. This is a question for Shawn from Sandra Encolada. Fantastic talk. She starts, have you applied your approach to do spatial mass spec of proteins in addition to metabolites in the HD brain? If so, with what labels?
We haven't done protein work. Our platform is really suited for small molecules, but the closest thing we've done is, and we continue to do, is we don't have full proteome coverage, but we have multi-IF. So we have about a panel of 30 different markers that we are starting to use for looking at 30 different proteins. But yeah, it's a different procedure for proteomics.
So this is one, Osama, not related to what you talked about today, but I know you're very well suited to answer this question. So this is from an anonymous attendee. Any comments or insights about the new theory that glial cell synthesis and release neurotransmitters and how this could be used for HD research? In other words, could looking at glial cells and modulating the neurotransmitters potentially slow down HD progression?
This is a great question. This is essentially the center of my research program, is to first understand how glial cells respond to disease, then use that insight to develop new ways of slowing down the disease progression. And with glial cells, we know that they are affected in Huntington disease. We know, for example, that oligodendrocytes, the cells that provide insulation to the neuronal axons, the extensions of the neurons that allow them to communicate. We know that these cells are abnormal. We know that they are in effect less able to perform their functions. And we know that astrocytes, which are the main support cells in the brain, are also abnormal. And relating to neurotransmitters, astrocytes do the basic part of, or the bulk of the housekeeping in sweeping away any excess neurotransmitters. And when astrocytes become abnormal with disease progression due to an injury, their ability to perform these housekeeping tasks is compromised, which makes the environment around the neurons relatively more toxic because it has a lot of neurotransmitters that may lead to neuronal death. So astrocytes and microglia and oligodendrocytes are affected in Huntington disease, and more work is now being done to understand how they are affected and use these cells as potential targets to modify disease progression.
Thanks, Osama. We have a question here from John Stevens that I think I'll take. He says, donating your brain for research sounded extremely important and something I hadn't really thought of. What's the best way to make people aware of this so more research can be done on the brain? I think there are several organizations that organize things like this. This is something that we actually had a conversation about. This webinar sparked an internal conversation about how we can get this information out there. So John Stevens, I am happy to send you some literature that we put together and get that information to you. But it's something that's not widely available. We definitely need better resources in the HD field, but these are very, very precious samples that scientists can learn so much from. And it is incredibly important. So this is a question for you, Shawn, from Herwig Lang. Do you find that in HD mouse brains, lactate is taken up instead of being released?
We have not used the lactate tracer in the HD brains. In the normal brains, we see it's a very small contribution to the overall carbon pool, but that doesn't, lactate is high on the list of, we have about 5 to 10 priority tracers that we're doing in the next wave. And this is really depending on, mouse availability, honestly. But the lactate is maybe in the top 3 even. So hope to update soon. Excellent.
Stay tuned, basically. So this is a question for each of you from Erica Villanova. For Osama, she says, thanks very much for 2 very nice presentations. For Osama, are individual glial cell types able to be investigated using spatial transcriptomics, i.e. astrocytes versus microglia or oligodendrocytes?
Yes, there are. So first of all, I presented one technique that allows us to perform spatial transcriptomics or measure gene expression in a spatially mapped fashion. And the technique that I presented does not get down to the resolution of a single cell. That being said, we can apply computational techniques to deconvolve or disentangle the contribution of different cell types in a specific capture area to gene expression to get down to the single cell level. That being said, there are also other technologies that allow you to measure gene expression at the single cell level. So the most widely available technique, which I touched on, Visium spatial transcriptomics, is commercially available, and thus far it's not single cell level or single cell resolution, but computational approaches can get you there.
Thanks, Osama. So Erica also has a similar question for you, Shawn. She says, is there a current way to extrapolate single cell resolution using imaging mass spec? Also, is there interest to look at gut differences in HD using this method?
So the first question, the technology is there for single cell, but it really depends on how you define single cell. So can we measure a 5 micron, a 10 micron spot? Yes. Are you shooting the laser at the direct nucleus in the center? So single cell to me means multiple measurements from a single cell. But to others, maybe 5 micron is the definition. But in any case, we're very close. I think MALDI will struggle to get down to 1 micron spatial resolution. I think 5 is well within reach, and certainly, we're doing 10. So we're very close. There's computational methods as well that merge H&E measurements and can make predictions that we've used, and you can actually get interesting resolution there. So yeah, I think the future is very bright, and 1 to 2 years before we see, good measurements. So, I mean, right now we can see things that ionize really well, but the sensitivity right now for small molecules, especially, which is really what we worked a lot with the company Bruker, which is the 9.4T magnet. We really dialed into 70 to 500 Daltons. So that's really like, glucose is, glucose and amino acids are in that range. So.
Thanks, Shawn. This is a question I think I'll send to Osama from Tacey Fox. I've heard that for an unspecified number of years, neurons with the HD aggregates that are, quote, sick can be rescued. Assuming an individual with under 45 repeats, do you think that an HD patient's neurons in the basal ganglia are able to be, quote, rescued after age of onset?
So first, a disclaimer, I am getting into the field of neuroprotection. So how do we protect neurons that are undergoing damage, continuous damage due to neurodegeneration? So neurons, like other cells in the body, they respond to damage by first increasing their the function of the machinery that allows them to fight that damage. And that damage in Huntington disease is manifest in different ways. And one way that happens is through protein aggregation. And one way a cell can fight that damage is to try and fold the unfolded protein to make the protein that is aggregated less aggregated. But then a cell can reach a limit, and that limit is determined by intrinsic factors in the cell and extrinsic factors. And in theory, a cell can be rescued. Now, what is the threshold at which a cell can be rescued? I think that's a topic of ongoing research, but in theory, a cell can be rescued before it commits to programmed cell death.
And I would add to that, there's newer research from Steve McCarroll suggesting, although not yet showing, it's very new stuff, showing that it's actually the Huntington repeats at the very end of a cell's life that are causing the toxicity. It's build up until that point that's really just increasing the sensitivity of the cell to its environment. And so on that theory, if we can regulate the extracellular environment, the environment that the cell is existing in, and make it more habitable for the cell, in theory, it should be able to be, quote, rescued. So it's interesting. This is one that will be aimed at Shawn from Jonathan Munkemeyer. Thank you for doing this research. Are you finding that a ketogenic diet is healthier for the HD mouse brains? And this is several parts. So maybe we'll start there and then we can answer the other ones.
I was just typing a response to that one. So, so yeah, we haven't systematically used a ketogenic diet in the HD mice. There's significant data on using ketogenic diet in HD mice. We definitely plan to revisit it using our technology, and I think we can learn quite a bit. There's another question about the conservation between ketogenic diet across different diseases, and I think understanding the mechanism of why these diets are effective in the first place could actually, lead to important information. But yeah, hopefully.
So there's second and third parts to this question. Are you finding that muscle wasting occurs with the enhanced glucose usage?
Yeah, so we'd love to be the ones to make this connection in a spatial way. I think the preliminary data definitely suggests that the muscle is burning more glucose, and when you have higher glucose consumption, you could wind up making more reactive oxygen species, and that could be bad for the cell. There's a lot of information about why muscles degenerate in the mouse models, especially so the microsatellite cells being impaired. And I think seeing this all spatially will hopefully shed some light. So we have nice markers where we can see type 1, type 2 fibers and different muscle cell types within. And so hopefully we can track down high levels of glucose consumption with cell type. That would be the next step for this.
And then lastly, the questions related to uric acid. Are the uric acid levels higher in HD because of higher glucose levels? And is the uric acid causing insulin resistance?
So as far as I know, it's not really known. I think it's very interesting and important phenomenology. And as I mentioned, in the sheep especially, you see this. So it's a mouse phenomenon, it's a sheep phenomenon. I imagine, I don't know the human data, but in any case, it's definitely important to figure out why it's higher, if it accumulates to a level that's toxic, and if there is any point of intervention, certainly suggests some nitrogen imbalance as well. So I think that's actually a really good avenue, and the subject of the sheep paper is great in 2017 PNAS.
So there's a question from an anonymous attendee that I think I'll take. It says, can you take a guess at when a therapy or treatment could happen? And I know this is the #1 question that families want to know. And unfortunately, it's an unanswerable question. No one has the answer of when this will happen. We all know we need it many, many years ago, and we all want it very, very badly. But what I think is important to highlight is the fact that even compared to 5 years ago, the number of clinical trials that we have, the fact that we're testing drugs in people with HD now, we are at a place in the HD research landscape that is at the frontiers of what we could have expected just a few short years ago. And so I think there's a lot of hope in that. And I think there's a lot of hope in what these types of techniques can tell us about those drugs. And so unfortunately, it won't be tomorrow. And it certainly wasn't 20 years ago. We all want it desperately, but it's incredibly exciting time because we actually have therapeutics being tested.
I'd like to add to that too, because I think the scope of our work, we look at, we talked a little bit about diet and nutrition, and these don't need, big, long approval processes that take forever. So while they might not be curative, if we can start to, better understand how they are causing problems, there may be some dietary suggestions that could take place, but we have to be very careful about this because it's easy to go down the wrong path. So, but I think we have the power to start thinking about these strategies.
Thanks for that, Shawn. Yeah, I think it's a point I think I pretty much touch on every webinar, but taking care of yourself, eating healthy, and diet and exercise really can do wonders. So thank you for sharing that point. It looks like we're at the end of our time. So if you have questions that didn't get answered, I will share those with our webinar speakers today. And if they're willing, they will get back to you. So I want to thank our webinar sponsors again, Neurocrine Biosciences and Unicure, as well as everyone who has donated to HDF. Our audience, thank you all for tuning in. And last but certainly not least, our speakers. Thank you so much, Osama. Thank you, Shawn. Registration for our May webinar is now open. We're going to be hearing from an HD researcher who studies sheep, Dr. Jenny Morton, as well as an HD clinician, Dr. Diana Rosas, about how sleep is affected in HD. So please sign up now, and you can join us next month for our webinar titled Counting Sheep, Does Huntington's Disease Affect Sleep? Thank you all so much for joining us today, and we hope to see you next time.